Welcome back. So in the previous video, we have already discussed about the process of DNA replication. Although we said that the process is highly accurate, but there are chances that a polymerase, it might incorporate a wrong nucleotide during the replication. So if you see that as per the Watson and Crick base pairing, we know that adenine, it will pair with a thymine, and similarly a guanine that pairs with a cytosine. But when a mutation is introduced, so you can see that adenine has to pair with a thymine, but instead a G is incorporated here. So during the second round of replication, when this strand is replicated, you will see that A will pair with thymine, but this G, it will pair with the cytosine. So the change from AT to GC will lead to a mutation in the DNA, and that mutation which is caused in the DNA, that can have deleterious consequences. Now, as we have already discussed before, that now if there are mechanisms in a cell to detect wherever there is this damage or wherever there is this misincorporation of the nucleotide bases. So now there are two things. One is the cell has to make sure it has to scan the genome. It has to see wherever there are any chances of any errors. So first thing. Then second thing is it has to make sure that it has to find out a particular machinery so as to repair those damages in the DNA. Now, if you remember in my previous video, we have already discussed about these methylations at the certain sites, that is the GATC sequences. These sequences, palindromic sequences, are present throughout our genome. We discussed them in the context of the origin of replication. And now this mismatch repair system, it basically makes use of this methylation marks to differentiate which one is the parental strand and which one is the newly synthesized strand. So here you can see that the parental strand, it will be methylated by the dimethylase on adenine, but the newly synthesized strand, it's not yet methylated. So this is a hemimethylated condition. So in this condition, the mismatch repair system takes advantage of this hemimethylated state, and it will see whether there are any misincorporations in the newly synthesized strand, and it will repair it. Now let's try to understand the mechanism of the mismatch repair system. So first and foremost, we have a dimeric protein, that is mut S. So as I said, first and foremost, what the machinery has to do is it has to ensure, it has to scan the genome in order to see wherever there are mutations. So first of all, it will scan the genome, it will scan the DNA, and once it encounters a kink, that means there is a misincorporation. So there will be a conformational change, and ultimately this conformational change, it will lead to the recruitment of the another proteins, that is the mut L and mut H. Although most of the reports have yet not elucidated what is the exact function of the mut L, but the mut H, one function is that it will come and it has both the helicase as well as the exonuclease activity. So it will create a nick, and then exonuclease activity, it will go on digesting this strand as long as also along with the with this nick. So afterwards, then we have the DNA polymerase. DNA polymerase, it will DNA pol third, it will come and it will carry out the synthesis, and eventually the ligase, it will seal the nick. So that was the process of replication. So that means there are three proteins, mut S, mut L, and mut H, which carry out the repair process. Again, there's one important thing, that is if the nick is created by the mut H on the 5' end of the mismatch. So you can see that here. So first and foremost, the mut S, it will bind to this site. So this represented as a kink, that is basically a mismatch. Now, if the nick is created by mut H on the 5' end, then we have the rec J or exonuclease here, which move from 5' to 3' direction. And eventually, again, the polymerases and the ligase that will then join it. Similarly, if the nick is created on the 3' end, so you can see nick is here, and on the 3' end of the newly synthesized strand, we have the nick. Then it will go on degrading the DNA, it will go on performing its exonuclease activity in this direction. So that is from here to here, along with this misincorporation of the base pair. So it will remove it, and then the polymerase, it will come and it will carry out the synthesis in this direction, 5' to 3', and eventually the ligase that will seal the nick. So that is how the mismatch repair system it exists, and how it basically repairs wherever there is a misincorporation, so as to prevent the damage later on. Now, similarly, we have multiple repair systems rather in our system. So we will discuss them one by one ahead as well.